A sperm whale died somewhere in the Pacific. What happened next lasted 100 years. The animal was 18 meters long, 40 tons. When the heart stopped, the body began to sink, slowly at first, then faster, falling through 3,000 meters of cold darkness, no light, pressure building to 300 atmospheres. It landed on gray abyssal sediment, and the floor shook. Within 2 hours, the first animals arrived. Craig Smith at the University of Hawaii had been waiting for this moment since 1989, when his team deliberately sank a whale carcass to 1,674 meters in the Santa Catalina Basin. What they documented changed deep-sea biology permanently. Not a slow decay, a succession, ordered and architectural. The first wave came fast. Hagfish, sleeper sharks, mobile scavengers drawn by chemical signals drifting upward through the water column. Somniosus pacificus, the Pacific sleeper shark, moved in slow arcs, unhurried. The soft tissue was gone within months. But that was only the beginning. By 2005, researchers had catalogued over 400 species across whale fall communities in multiple ocean basins. Smith's 1,989 Nature paper identified at least 30 species that appear nowhere else on Earth. Creatures that exist only because a whale died in the right place at the right depth, specialists, obligates, animals that evolved around a carcass. The bones themselves became the next ecosystem, not a remnant, but a resource. A single 40-ton gray whale carries 2,000 kilograms of lipid stored inside its skeleton. That's enough chemical energy, according to Smith and Amy Baco's 2003 review in Oceanography and Marine Biology, to sustain microbial communities for 50 to 100 years. One body, 1 century of life, in total darkness. What happens inside those bones will rewrite where the deep sea's most extreme ecosystems actually come from, the specific chemistry, the organisms that evolve to unlock it. That answer hinges on a single biological process that begins not on the surface of the skeleton, but inside it. Craig Smith's team at the University of Hawaii didn't stumble onto whale falls. They built a science around them, systematically, over decades. February 1987, the submersible Alvin descended to 1,240 meters in the Santa Catalina Basin and found a whale skeleton already in progress, bones white against black sediment. A community already established. Smith recognized it immediately. This was not random, this was a system. By 1989, his team was deploying carcasses deliberately, sinking a whale to 1,674 meters off California and watching what arrived. The data produced a 4-stage model: clean, elegant, peer-reviewed. Stage 1 brings mobile scavengers. Sleeper sharks, hagfish, rat-tail fish strip 40 tons of soft tissue in months. The bones emerge bare, pale, almost surgical. Stage 2 brings enrichment opportunists. Smaller invertebrates colonize the exposed surfaces. Polychaete worms, crustaceans, mat-forming bacteria. Hundreds of species move through in overlapping waves. Stage 3 is the sulfophilic stage. Bacteria penetrate the bone itself, consuming 2,000 kilograms of stored lipids. Smith and Amy Baco documented this in 2003. Their words, "The lipid-rich bones of a single large whale may sustain a sulfophilic community for decades." Stage 4 turns the skeleton into a reef, hard substrate colonized by suspension feeders. The skeleton becomes architecture. 4 stages, documented, reproducible across ocean basins. By 2005, researchers had catalogued over 400 species from whale fall communities worldwide. Smith's 1,989 paper in Nature confirmed at least 30 appeared nowhere else on Earth. The model holds until stage 3. The organisms that arrive during the sulfophilic stage, the bacteria, the limpets, the tube worms, have closest genetic relatives that do not live at whale falls. They live at hydrothermal vents, isolated, thousands of kilometers away. Robert Vrijgenhoek at Mbari confirmed the evolutionary relationship in 2004. The species are nearly identical. So, if vent species evolved at vents, how did they reach this whale? Here is where everything the whale gave becomes permanent. In 2003, Craig Smith and Amy Bacco published their findings in Deep Sea Research 1. 11 species recovered from whale fall sites had also been documented at hydrothermal vents and cold seeps. Chemosynthetic invertebrates with no other known habitat anywhere on the ocean floor. 11 species, nowhere else. Only whale bones and volcanic vents. Smith and Bacco proposed something that rewrites the map. Whale falls are spaced, on average, every 5 to 16 kilometers across the Pacific floor. That distance is close enough for larvae to drift between them. Each decomposing carcass functions as an island, a biological waypoint between vent fields separated by thousands of kilometers of otherwise barren abyssal plain. The bone lipids make it possible. A single 40-ton gray whale carries 2,000 kilograms of lipid stored inside its skeleton alone. When sulfate-reducing bacteria metabolize those lipids, they produce hydrogen sulfide, the same chemical currency that powers hydrothermal vent ecosystems. The chemistry becomes indistinguishable. The species that arrive are genetically traceable back to vent populations. Smith and Bacco wrote it plainly: whale skeletons may provide stepping stones for the dispersal of sulfur, reducing bacteria and their animal symbionts in the deep sea. That process sustains a chemosynthetic community for 50 to 100 years per whale. A whale dies, it sinks, bacteria colonize the bone, hydrogen sulfide rises from the sediment, vent specialists arrive, the same organisms that live beside volcanic fissures at 2,000 degrees. They reproduce, their larvae drift. They find the next whale, then the next vent. The deep sea's most extreme life, the life that survives without any sunlight, without any photosynthesis, at pressures that would collapse a submarine, may have spread across the entire Pacific floor by traveling through the dead. What the index concludes: a whale does not simply die, it becomes infrastructure.